Okay, so students, in today's lecture we will be studying about the advanced Bell stage and root formation, how the root is formed. So first of all, we are familiar, this was the enamel organ and this is the dental papilla. There is the signaling of the inner enamel epithelial cells to dental papilla cells, and they are going to convert into the odontoblasts. Alright, so if you are asked about the advanced Bell stage, you are going to write the three important points. The number one, it must be the mineralization is taking place in this stage, and number two, there is the root formation happening, and number three, there is the determination of the dentino-enamel junction. So later I will be telling you about the dentino-enamel junction. So advanced Bell stage, in the advanced Bell stage, these are the three events they're going to happen. So what is the dentino-enamel junction? So as the name says, the junction of the dentine and the enamel. So the outline between you have to write the proper definition of it. So the outline that is between the inner enamel cells and odontoblasts. Alright, this is the dentino-enamel junction. So these were the inner enamel cells, and these are the reddish ones are the odontoblasts. So the outline that is forming here between the inner enamel cells and between the odontoblasts, this reddish layer is known as dentino-enamel junction. Alright. So after the formation of dentino-enamel junction, the odontoblasts, these reddish cells, these odontoblasts, they start forming the dentine. So the dentine, they are going to start the formation of dentine, and that dentine is known as radiculodentine. The dentine that is covering the root is the radiculodentine. So this is the structure of our tooth, and this is the root of the tooth. So we have divided this whole thing into three parts. This is the coronal, upper one is the coronal portion, or the crown one, having the crown. This is the middle portion, and this is from this to this is the apical portion. So whenever there is the activity of the odontoblast, suppose here are the odontoblasts, they are going to form the dentino-enamel junction. From the junction of hair, from the enamel junction, they will start forming the dentine towards the apical direction. So whenever you are asked how the odontoblasts are going to form the dentine, they are always going to form the dentine in the apical direction, or the downward direction, towards the apex. This is the apex of the root. Alright, so wherever there is the formation of the apex, towards the apex, okay, fine, the only thing you are going to remember is apically there will be the laying out of the, or the laying down of the dentine. So now coming up to the next diagram, when the dentino-enamel junction is determined, this reddish portion, the inner enamel epithelial cells will give rise to the enamel, and these odontoblasts are going to form the bluish layer or the dentine. Alright, this upper layer is enamel, and the lower bluish one is dentine. So coming up to the next thing, what's happening, there is a very important thing, that is the enamel diaphragm. So let's make a concept of this enamel diaphragm. Suppose we have this inner enamel epithelial layer, and this was the outer enamel epithelial layer. Whenever the inner enamel epithelial layer or the outer enamel epithelial layer, they both are joining, they are going to form another layer. These two layers are going to join the inner enamel epithelial layer and the outer enamel epithelial layer. They are going to join, and they are going to form a diaphragm, that is the enamel diaphragm. Alright, so whenever you are asked about the enamel diaphragm, you are going to write that inner enamel epithelial cell layer and the outer enamel epithelial cell layer, inner were the columnar and the outer were the cuboidal, they are joining, and joining to form what? An enamel diaphragm. This is a third layer formed by the combination of these two layers. So what is the function of enamel diaphragm? We will be studying it later. So what's happening? So the thing that is, once the dentine is formed, so after that there will be the differentiation of the inner enamel epithelial cells into what? Enamel. But now we are getting into the detail, how the series of events are happening here. Okay, so suppose this is the innermost layer or the pulp of the tooth. This bluish outer layer is the dentine. Then this darker enamel, this is the enamel, and outside and when we are going to subdivide the enamel, we are going to read the details of the enamel. In the enamel there is the inner enamel epithelial layer. In between the inner enamel epithelial layer and the outer enamel epithelial layer, we have the stellate reticular cells, these star shaped cells, or the cross I have formed in this diagram. And here you can clearly see in the cross sectional area or the cross sectional view, you can see the outer enamel and the inner enamel are going to form the enamel diaphragm. So now the story begins here. The root formation begins here. How the root formation is going to take place. Okay. So this is, suppose this is the enamel diaphragm. This was the inner enamel epithelial inner one layer, and this was the outer enamel epithelial layer. Suppose we have overlapped these both layers. So what's happening, this inner enamel epithelial layer, this layer induces the formation of the radicular dentine. Alright, this is the formation of what? Inner enamel epithelial layer induces the dental papilla cells, and they are going to form the odontoblasts, and these odontoblasts are going to form the radicular dentine and the dentine that is present on the root surface. The arrows I have formed, this is suppose the radicular dentine. Alright, and the outer enamel epithelial layer, they are going to form the epithelial root or the Hertwig's epithelial root sheath. Alright, so this is Hertwig's epithelial root sheath. This is the sheath that is formed and helping the formation of the root of a tooth. Alright, so now coming into the more details, whenever, suppose this is the formation of, okay, I'm going to make it more easier for you guys. Okay, so suppose this was the radicular dentine outside and this was the Hertwig's epithelial root sheath. Whenever there is the break in the continuity, the breakage in the continuity of the Hertwig's epithelial root sheath in the periodontal ligament. So whenever there is a breakage in the continuity of Hertwig's epithelial root sheath, there are a few epithelial islands or the remnants. First of all, we have studied previously there were the epithelial rest of Ceres in the dental lamina, but no, now we are going to study the epithelial rest of Melissus. So these epithelial rests or the islands of the epithelial cells, those were the Hertwig's epithelial root sheaths. They are formed inside the periodontal ligament or the periodontal space or the periodontal tissue, the supporting structure surrounding the root. These are called the epithelial rests of Melissus. So these are what are the rest of Melissus? These are the remnants of Hertwig's epithelial root sheath in the periodontal space or the periodontal ligament area surrounding the root structure. So these are the clumps or the epithelial small epithelial islands. Alright, here you can see. Okay, now coming to our point again. So students, as you all know, this was the enamel organ. Now here is what? Epithelial root sheath. Outside there is the formation of the radicular dentine. These arrows I have formed, this is the radicular dentine, and inside there was the Hertwig's epithelial root sheath. Outside here is the radicular dentine, and here you can see This was the dental follicle or the dental sac that was surrounding the structure, enamel organ and the dental papilla, the supporting structure in the connective tissue. This was the connective tissue. So the dental follicle cells, these blackish ones are the dental follicle cells. These cells are now going to interact with the radiculodentine. Whenever they are going to signal or interact with the radiculodentine, okay, so the dental, for what is happening, I'm going to make a flow chart. The dental follicular cells, they are going to interact with the radiculodentine. The dentine that was formed by induced by the inner enamel epithelial cells or by the odontoblasts. Alright. So whenever there are the dental follicular cells in the dental follicle, they are going to interact with the radiculodentine. They are going to form the cementum. Remember, students, cementum is the outermost layer present on the root structure. Cementum is absent on the crown structure. Cementum is only present in root, and there is not enamel that is present on the root structure. There is no enamel on the root structure, but there is the cementum on the root structure, the hardest tissue present. So this is how the cementum is formed. Okay. So these bluish cells I am going to show you are the cementoblasts. And these cementoblasts are going to form this layer outside the root radiculodentine, the bluish layer, this bluish layer. This is what? This layer is cementum. I hope you are clear now. Okay. So let's just go to the detail of how the roots are formed. Suppose these are the mandibular or the lower molars. Okay, now let's go back to an idea. This was the enamel organ and the enamel diaphragm. Alright. This is the cross-sectional view of a root. There are the two enamel diaphragms made up from the outer enamel epithelial layer and the inner enamel epithelial layer. They join to form the enamel diaphragm. Whenever on the surface of the root there are the two enamel diaphragms formed, they are going to join with each other, and whenever they are going to join to each other, and then they are going to split and two roots are formed. Alright, that's why the lower molars or the mandibular molars have two roots. Alright, these are the mesial or the distal roots. You will be studying it in tooth morphology. Okay, and now coming up to the next thing, upper molars or the maxillary molars. There were the three enamel diaphragms, they were formed, and they are going to split into the three roots, and we have the mesiobuccal, we have these mesial roots, and this is the mesial, this is the distal, and this was the palatal root. So mesial root is basically what? Mesiobuccal or distobuccal, and we have this palatal root that is towards the palate in the upper molars or the maxillary molars. So here are a few anomalies or the loopholes in the formation of this whole bell stage. So whenever there are two things inappropriately happening. Number one, that these are the enamel pearls. Number two, these are the accessory canals. How the enamel pearls and how the accessory canals are formed? First of all, students, there shouldn't be any enamel on the root structure. This is a note. Whenever there is enamel on the root structure, that is something inappropriate, an anomaly, or something bad. So if the Herdwick's epithelial root sheath persists, it's not going to dissolve, not going to absorb. Then a few islands are formed. These islands will signal the inner enamel epithelial cells to convert into the ameloblast, and then enamel is formed. These are going to form the enamel pearls or the enamel stones. Mostly the enamel pearls are located on the furcation area. This is very important for your MCQ. Furcation area, the area that is between two or at the junction of two or three roots. All right. So mostly these are formed at the furcation area. So when we are going to study about the accessory canals, how the accessory canals are formed. So this was the Hertwig's epithelial root sheath. Whenever the Hertwig's epithelial, there is any breach or breakage in the continuity of the Hertwig's epithelial root sheath before the laying down or before the formation of the radiculodentine. Before the formation of what? Radiculodentine. We have this accessory canals are formed. Accessory root canals are formed. Alright. So these are the things you guys are going to remember. These are with the persistence of the Hertwig's epithelial root sheath, we have what? We have the enamel pearls or enamel stones, mostly located at the furcation area, and at the before the time in the breakage of the continuity of the Hertwig's epithelial root sheath, we have what? We have the accessory canals formation. So this was all about the advanced bell stage, and now in our next lecture we will be studying about the mineralization and the morphodifferentiation.